All right, everybody, welcome back. We are continuing on to Module 7.1, where we're going to be discussing eukaryotic diversity. I apologize for any confusion that we had this week. I didn't realize that Indigenous Peoples Day was during Monday, and and unfortunately, I had to go back to the hospital. But I'm back now, and I'm hoping that I will be home for the remainder of the semester. And I'm, as always, I'm available by email to sort of sort any questions that you guys might have out. And I additionally putting on a couple extra opportunities to meet this week. So previously we were discussing prokaryotic diversity, and we had sort of made the jump into eukaryotic diversity by starting to talk about plants. Well, today I wanted to start off with a study that sort of is an interesting study in that it encapsulates a lot of the different organisms that we've been talking about. So this is a study by Dr. Naima Harris looking at species loss on spatial patterns and the composition of zoonotic parasites. So essentially what she's looking at is she's looking at apex predators and the compositions of parasites that these predators carry. And these parasites that these predators carry span all different types of organismal biology. We have bacteria, which again, prokaryotic, which we've discussed previously. We have protists, which we're going to be talking about today. And we also have things like helminths and roundworms. And then ultimately, we then have the host. So these are all taking place inside of apex mammal species of the family Chordata. So that's what we're going to sort of be talking about today is how these different organisms mutually survive one another and what distinguishes each of the organizations of these different types of phyla. And specifically, what Naima was looking at was if certain apex predators go extinct, how does that impact the population of these potentially disease-causing helminths, bacteria, and protozoa? So this is just an interesting study that's looking at all of these different aspects. So the driving questions that we're going to be looking at for this module is going to start with what exactly are eukaryotic organisms and what factors influence their diversity? How are animals defined? What influences their diversity? How are fungi defined and what influences their diversity? And lastly, how are protists defined and what influences their diversity? So a lot of these lectures are going to be very definition based. It's going to be understanding how eukarya is organized and what specific characteristics influence that organization. So as we've done in the previous two lectures, let's center ourselves in our phylogenetic tree where we have our tree of life, and we have bacteria, archaea, and now we've jumped into eukarya. So we talked a little bit in our last lecture about plants, which are a multicellular eukaryote. They have cell walls. They're photosynthetic, so that means they're making their own food, or they're considered autotrophic. And then they're adapted to life on land. And we talked a little bit about the organization of plants, specifically looking at going from non-vascular to vascular plants, and then looking at the development of seeds that go then into gymnosperms and angiosperms. Well, today we're going to be talking about, broadly speaking, animals. So when you think of an animal, you probably envision something like what you had in your book, where you have these kingfishers or these or something like a banana slug. But I'd like to just go ahead and take a minute and think of an animal in your mind. Now, the critical things that sort of define animals broadly, keep that animal in your mind, is that it's a multicellular eukaryote. and then it obtains its nutrients by ingesting other organisms. So in this case, it's a heterotroph. Now, go ahead and put a finger down if your animal had fur. Put a finger down if your animal had a spine or a backbone. Put a finger down uh, if your animal was a bug or a fish. So odds are you were probably envisioning what we sort of think of as what are quote unquote like the most relatable animals on Earth. However, the vast majority of life on Earth, in fact, over 75% of life of animals on Earth, fall into other categories. And that's what we're going to be talking about today. So to start off, we're going to sort of be looking at a very broad way of categorizing animal life, and that's just simply into vertebrate and invertebrate animals. So whether or not an animal has a spine. And looking at this slightly more branched out family tree, we can see that the vast majority of animals in the family Eukarya do not have a vertebra. And really, we're only talking about chordates that have a vertebra. So again, how do we define invertebrates? It basically just means it's grouped based on lack of backbone. And really, and that group alone encapsulates 95% of all animals. And those can be broken down into mollusks, annelids, flatworms, nematodes, arthropods, and echinoderms. And then vertebrates, where we're starting to talk about chordates, are animals with a bony or cartilaginous backbone. And again, these are all chordates have a vertebra. The second broad way that we're going to start to think about how biologists classify different types of animals is looking at a concept called symmetry. So at its most basic, we have sponges, and you can see that there's no symmetry in the body structure of a sponge. However, if you look more at like corals or polyps, you can see they have something called radial symmetry. So if you drew lines through this, you'd be able to get symmetrical sections of the animal. And then lastly, we have something that's called bilateral symmetry, which is really saying we can split an animal in half and they're going to be equal on either sides. Now, just a reminder that ultimately when we're thinking about these phylogenetic trees, at the end of the day, the interrelatedness of different species of animals is based on their genomic makeup, not necessarily on characteristics such as body plan. This was, in fact, sort of an old way of how we classified biology on Earth. But there are still similarities in broad classes such as vertebrate or invertebrate that seem to stand up fairly well in terms of the taxonomy by genetic affiliation. So again, we can really sort of think of some of these large, broad classifications of life on Earth. So we had our first one was vertebrate and invertebrate, and now we're starting to think of these three main lineages of symmetry. So in looking back on what is most recently diverged from the common ancestor, we have things that have no symmetry. We have things like sponges. And these are really the simplest living animals. And they lack defined tissues or organs. And then as we start to go up our tree and farther away from our common ancestor and branch out, we start to see things like radial symmetry. And this is when we get nidarians, which are like jellyfish, a lot of aquatic life, a lot of coral life. And I have a specific video for you guys to watch on nidarians because they really are the start of a lot of different of different evolution that we start to see such as movements and digestion, that obviously are very important for everything else down the line. So again, Nindarians have body plans that are circular. They have that radial symmetry. There's no clear left or right sides. And as we start to make our way up, we start to have organisms that have bilateral symmetry. So they have clear left and right sides, and they're ultimately mirrors of one another. And this is where we're going to start to pick up in our next lecture, is starting to, now that we've gone through sort of the very simple formation of life of sponges and Nindarians, to these more complex organisms such as flatworms, mollusks, and annelids. So that's where we're going to pick up next time. So see you soon.